We should be recording now. Excellent. Well, hello everybody. It is great to see you all as always. And today we are diving into potassium. And two articles for today. The main one, I think, is this newer review. The other paper that I had sent out, which was the potassium homeostasis and renal failure, I think was okay, but it actually was a little bit disappointing to me, partially because it was older and a lot of the studies that were cited were really coming from two people with an oliguric kidney disease, and it was just a bit annoying in that regard. I think we'll probably spend most of our time focusing on this paper, and that should be okay because you know what? We have a lot to get through. This is a pretty big paper talking about potassium physiology and regulation. So let's dive into it. Anybody have any questions from the reading that you want to jump to first? You can see I have a lot of stuff highlighted, so we have a lot of stuff that we can get through. All right, well, as always, feel free to unmute, uh, raise your concerns, ask your question. Let's make this a discussion rather than just a one-sided lecture on my end. But I thought this was a nice way. We've been talking about sodium, we talked about diuretics, we talked about the SGLT2 inhibitors and how they would potentially affect GFR and urine output and the effects they might have, which are somewhat due to inhibiting the absorption of sodium. Remember, the SGLT2 inhibitors reabsorb a molecule of glucose and a molecule of sodium. So now we're flipping towards potassium. And I thought this paper was really nice. It was pretty thorough, talking about things other than the kidneys, but also just very important in terms of just the overall regulation of potassium. And let's get into it. Let's see what we got. So starts with just some of the regulation of potassium balance. And we remember that potassium is our major intracellular cation, and the concentration of potassium and sodium are pretty much opposite intracellular and extracellular. So the intracellular potassium concentration is almost around 140 or so, very similar to the plasma concentration of sodium. The intracellular sodium concentration is usually around 5 to 10. It's pretty darn small. And the reason for that is we have this basolateral um, sodium potassium ATPase, and that's what's responsible for keeping your potassium in the cell and your sodium outside the cell. And so that uh, transporter is really very, very responsible for keeping that internal and external environments different, as well as establishing the gradients for sodium to enter the cell, which many things are tacked onto in terms of utilizing that electrochemical gradient to be able to perform secondary active transport. They talked a little bit about catecholamines and insulin being the main regulators of moving potassium inside the cell, and those are going to be done through two different receptors. So we have our GLUT4 receptor, which is where glucose will bind. And so insulin has a different receptor. Insulin phosphorylates the intracellular messenger, the PI3, which leads to insertion of these GLUT4 transporter channels on the cell surface. And uh, catecholamine, specifically through our beta receptor, will stimulate the entrance of potassium into the cells through direct stimulation of the sodium potassium ATPase. And we utilize this, right? This is not surprising. We use these mechanisms for the management of our patients with hyperkalemia. So giving insulin is going to stimulate this intracellular mechanism that not only will put more GLUT4 receptors on the cell surface, causing glucose to move intracellularly, which is why if we're giving insulin to treat hyperkalemia, unless they are hyperglycemic, you have to follow it with dextrose as well too. Otherwise, they will become hypoglycemic. But that messenger also stimulates the sodium potassium ATPase on the basolateral side, moving potassium inside the cell, and then giving something like terbutaline or a lot of albuterol will stimulate the beta 2 receptor and will cause activation of that as well. Now, are the effects additive if you use terbutaline and insulin and dextrose? Will that maximally activate that basolateral receptor? That's a good question. I don't know if anybody has actually done a study looking at the movement of potassium with one or the other or both of those therapies. We certainly use both of those therapeutics concurrently to manage some of our hyperkalemic patients. So it is, I don't know if it is additive. I think that it probably is, but I guess I don't know of a study that has specifically looked at that, but that study might exist for sure. So catecholamines and insulin, those are our main regulators of driving potassium inside the cell. And they talked about the effect of exercise, which is not so much a problem for our patients. We're not seeing many working dogs or sled dogs immediately afterwards, but I thought it was pretty interesting that interstitial potassium can be high as 10 to 12 with severe exercise. And from a personal standpoint, I once had to get blood work checked, and the day before I had my blood work pulled, I had ridden a century, so I rode a 100-mile bike ride the day before, and my doctor called me because my potassium, I think, was 6.0 or something, and she was very nervous and wanted me to go and get it rechecked, and I obviously felt fine, but I said, I just did this 100-mile bike ride yesterday. Is it possible that that could be playing a role? And she uh, was very fine with that being a reason and didn't care that I that went and had it rechecked. And then subsequently, my potassium has been normal. So that's the only time that I had it be abnormal. So that's an interesting effect that we can see with exercise. Something else that's been noted in people is how much you can have the your plasma potassium concentration increase with um, with a tourniquet use, and this is something I can see if I can pull it up. Uh, I don't even know if I'm definitely not spelling tourniquet correctly, but there are a couple papers that have looked at it, and it can increase your potassium by, by a couple points. Ugh, I'll have to pull it and then pull the papers and find it, and I'll send it out. Where a potassium of 7 has been seen in people when they're using a tourniquet to get blood draws, and some of that's probably from squeezing and the muscles and having altered blood flow. And it really reminded me of all these cats that we hold off the leg for so long before we're able to draw blood on, and you wonder what effect that might have and how many of our patients might have a iatrogenic rise in their potassium concentration because of it. So I'll try to pull those papers and just email it to everybody to have you add that to your library. So, and just an interesting factor. Okay, then they talk about acidosis, and here is something that, to be honest, I really had to try to dive a little bit deeper into this. Um, Before that, they talk about hyperglycemia causing um, hyperkalemia because of solvent drag. That is not something we see super commonly because the hyperglycemia also causes the osmotic diuresis, but you can see, and there's even a case report of hyperkalemia associated with a mannitol overdose. So if you give too much mannitol, it's going to pull water out of the cell, and that's going to draw the potassium with it as well. And you can have hyperkalemia associated with that solvent drag. So then they talk about acidosis, and they talk about mineral versus organic acidosis causing that shift in potassium. And this is something that I have read and I have memorized, but to be honest, is it something that I could explain? No, it wasn't. So I dug into it a little bit deeper, and it was actually really nice because they gave these figures here in figure 2. This is looking at the change in potassium with or a mineral acidosis or an organic acidosis. So the same players are involved, except for your organic acids, those are going to be able to be transported by this particular transporter, this MCT1 and MCT4. And that's your monocarboxylate transporter. And this actually makes all the difference in the world when it comes to the development of hyperkalemia associated with acidosis or not. So if we just review and we're talking about organic versus inorganic, so the minerals, those are your inorganic acids. That's going to be hydrochloric acid, phosphate, and ammonium. Those are the main ones. So clinically in our patients, we don't usually see hydrochloric acid in the blood. We don't usually see that. We can see ammonium be present, and obviously we can see hyperphosphatemia, but this is I think a very uncommon situation. When we see it, it should cause a normal anion gap hyperchloremic metabolic acidosis. Most of the acidosis we're going to see is going to be organic, meaning it's coming from lactate, from keto acids, from uremic acids. Most of those are also organic. So, this is, I think, a point that comes up often talking about acid base and potassium, but to be honest, I don't know how often we really see a true inorganic or mineral acidosis. But the difference is we have this monocarboxylate transporter, this MCT1 or MCT4, and that will actually bind molecules that have a carboxylate group, and those include molecules like lactate, pyruvate, ketones, and it will transport it across the cell. So now we have the difference where we have your conjugate base, so the lactate and the hydrogen ion both can get transported inside the cell, and that's going to affect the internal cell pH because we have all this extra hydrogen that's there, and that will change the efficacy of all of these channels, and we end up not having the potassium efflux that we do with a inorganic or a mineral acidosis because you're not having that accumulation of intracellular uh, hydrogen ions. So that's the big difference. And honestly, it wasn't really through going through this review and digging a little bit deeper that I understood why that might be the case. So I thought that was a nice eye-opening moment for me to learn more about the mechanism rather than just memorizing that hyperkalemia is associated with a mineral acidosis, but not an organic acidosis. It all comes down to this MCT1 and 4 transporter that's going to shuttle the hydrogen ion inside the cell. It's going to make the cell more acidemic, and that's going to change the movement of sodium across, and by secondary effects, it's going to change the movement of potassium. So that was a nice review. I was happy to kind of feel a little bit more comfortable with that. Is there any questions on that aspect? Does everyone else feel good about that? Am I just behind the 8 ball regarding that topic, or anybody else have any questions or challenges there?
Just a quick thing, JD. I appreciate you delving into this because this is something that's been bothering me for a long time too. I might have misunderstood it, but it almost sounded like both situations result in an increase in hydrogen ions intracellularly, but I think the last statement you said made it more likely that the organic acid is going to have an increase in intracellular hydrogen. Can you just clarify?
Yeah, and I definitely could have said it incorrectly for sure. Yeah, so because you have the transporter here that's going to pull the hydrogen ion and the conjugate base along with it, it will result in a higher intracellular accumulation of hydrogen ions. So you're going to have the intracellular environment will become more acidic with the organic acidosis than the mineral. Because you don't have that transporter, you still have the extracellular environment that's acidic, but you end up not having the intracellular environment that is so acidic. So you end up having more hydrogen and sodium that's shuttling in the inorganic model. So you'll have a change in your intracellular sodium concentration that's going to push the movement of some of these other um, transporters, and you'll have greater potassium efflux as a result. And because the pH of the intracellular environment is not so much changed, you actually will not have. Um, so let me say that one more time because I think I'm messing it up. With the inorganic acidosis, because the pH is relatively neutral, it's going to account for that change in the intracellular sodium concentration by shuttling potassium. So you get the hyperkalemia. With the organic one, because of this transporter resulting in the intracellular acidosis, it's going to change and buffer the cell through other mechanisms, and you actually don't have significant potassium efflux as a result of it. So it really comes down to shuttling that conjugate base along with the hydrogen ion inside the cell, which is only going to be with an organic molecule, again, that monocarboxylate and that MCT monocarboxylate transferase, you require the carboxylate group. So if your molecule does not have a carboxylate group, like hydrochloric acid, it's not going to be able to be a substrate for that transporter, and it will cause the acidosis, but you don't get that intracellular acidosis, which is what is the main determinant that's going to trigger the efflux of potassium.
Thanks, that helps. I'm going to have to modify my very simple hydrogen ions go in and potassium goes out
Yes,
as a general explanation. Just
yes. That's the way I've always thought, and it's so easy and beautiful. And of course, it can't be the case. It's got to be more challenging than that. That is not that simple. But to be honest, I just don't know how often we see this, even in our patients that have significant hyperphosphatemia. And you could say, okay, well, there is a component of a mineral acidosis from the hyperphosphatemia. Those patients also have uremic acids, and they might even have a lactic acidosis if they're hypoperfused. So they could have the organic acidosis too. So it's hard to know to what extent that could play a role. And certainly in our DKA patients, we're not going to be expecting that because that's all going to be organic and lactate, but it is not nearly as simple as too much H plus. That's going to go in the cell to maintain electroneutrality. Potassium goes out. That
um,
summary sounds fantastic, and unfortunately, it's more complex. But I thought this was a nice review, and they talk through it step by step in the legend to Figure Two. So I thought that was a nice description rather than just only stating, which so many other texts that I've read will do, just stating the difference in potassium, extracellular potassium concentration with organic versus mineral. So I really appreciated them getting into that so I could feel a bit more comfortable overall. Okay, let's see. What else? Okay, so if everyone's okay with that, we can move on to the renal potassium handling. This I think is the bulk of kind of what we're going to get into today, and I think has some cool features. So potassium is a very, very small molecule. So it's going to cross the glomerulus. So in a normal patient, most of the potassium in the early portion of the nephron, and let me actually say probably all the potassium in the early portion of the nephron is all from the filtrate. It's only in the distal portion of the nephron, and that's probably just the collecting duct, where the potassium in the filtrate is going to be secreted potassium. And that is true even when we're actually reabsorbing most potassium. You're probably going to reabsorb it all and then secrete a little. So it's not even so much that the little bit of potassium that ends up in urine is what was not reabsorbed. That's not actually quite true. You pretty much reabsorb everything and then you're excreting it. So there is very little that is excreted. Under normal circumstances, only 10% of the quantity that's filtered is ends up going out into the final urine product. But we have a couple different mechanisms on how potassium's handled. In the proximal tubule, it's easy because it's just following sodium. So we have active reabsorption of sodium here. Water is going to get pulled through the paracellular route and it will be absorbed into blood. So the absorption of sodium is going to be isotonic. So your tubular filtrate at the start of the proximal tubule, let's say your plasma osmolality is 300, your tubular filtrate uh, will have an osmolality of 300. After the absorption of the sodium and water, potassium is going to get pulled in with it just via solvent drag. In the late proximal tubule, your filtrate's osmolality will still be 300. So you're going to pull out solutes and water similarly, but about 2/3 of your potassium is going to be reabsorbed here, just like 2/3 of your sodium is reabsorbed in that proximal tubule. And it's a very passive mechanism that exists. So that's the bulk of your potassium reabsorption. Then there is a little bit in the ascending limb of Henle, and that is kind of from the ascending limb into the distal tubule. That's probably going to account for another 25% of your potassium. And this is where things get really, really interesting. And I will say that because I think it's also really confusing. Because what's happening here is the potassium is getting recycled. The potassium is coming in, then it's getting sent out, only to come in again to get sent out. So we have here our sodium potassium 2 chloride cotransporter. We all are very familiar with this. It's the main driver of reabsorption of sodium and chloride in the thick ascending limb. It's the site of action of furosemide. So here we have a uh, absorption of 2 positive molecules, 2 sodium molecules. We reabsorb that from the tubular lumen into the intracellular space. And then once we're here, we have the basolateral sodium potassium ATPase that's going to kick out that sodium. Water is going to follow through that. The potassium can leave through that some basolateral channels, but we also have this channel here on the luminal surface. So as that potassium starts to accumulate and the concentration begins to get too high, the mechanisms for it leaving includes this potassium chloride cotransporter, but we also are getting rid of that extra sodium. So we're having potassium getting drawn in by that sodium potassium ATPase. So the combination will lead to potassium accumulating in the intracellular space. So we have this renal outer medullary potassium channel. That's the ROM-K channel. And it will cause potassium to efflux out back into the lumen, where it becomes available to bind to sodium and chloride and to be shuttled back into the cells. And then it leaves again. So it's just looping around and around and around. And if you think about it, it makes sense. Even though we have reabsorbed a similar proportion of the filtered sodium and potassium in the proximal tubule. Let's say we've absorbed 2/3 of the sodium in the filtrate, we've absorbed 2/3 of the potassium. That is true. But if you look at the actual quantity of solutes, the sodium concentration in the proximal tubular lumen is going to be around 145. The potassium concentration is going to be 5. So we reabsorb a portion of that. Now when we get to the thick ascending limb, we still have so much more sodium than we do potassium. So if we did not have this potassium recycling, the activity of the sodium potassium 2 chloride cotransporter would shut off because we didn't have any more potassium left in the lumen. So you need potassium to do this. If you don't have enough potassium, if you're hypokalemic, then hypokalemia, the not enough potassium here, that could start to shut down or decrease the activity of that sodium potassium 2 chloride cotransporter, which is why hypokalemia can cause PUPD. It kind of works like Lasix. It's amazing. It's so cool. So it all has to come down to this shuttling, this recycling that's there. So potassium gets put in, then it just leaks out through that ROMK. It's available to then get picked up with more sodium and chloride, get reabsorbed, and go right back out. So that recycling is really, really important to allow for that sodium potassium 2 chloride cotransporter to work. But you still have some basolateral reabsorption, and that's going to be through also some potassium channels, and then also the you want to get rid of all that chloride that's accumulating. So that's a potassium chloride co-transporter that's going to shift it through that basolateral side. So I thought that was was really cool to see, and that recycling again made it clear why hypokalemia is just not on our list of differentials for PUPD, but this is why. Hypokalemia is essentially baby Lasix. If you don't have enough potassium around, you cannot have the sodium potassium 2 chloride transporter be effective. And that is pretty awesome. Does that make
JD,
sense?
do you know if there's a level of hypokalemia that would result in this? I would presume pretty moderate to severe.
Yeah, that's a really good question because you can imagine the difference is huge, right? 5 millimoles of potassium versus 145 of sodium. It is already a huge disparity between the two. But I don't know if anybody has worked out if you get to 3 millimoles, is that where you see it? If you have to be down to 2.5, I haven't seen that, and unfortunately, it probably would have to come from animal models because I think our patients who have naturally occurring hypokalemia, they're undergoing it because of some other systemic cause, like diabetes or CKD, that would cause PUPD in other mechanisms as well. So I haven't come across the magnitude of hypokalemia that is needed for that to occur. But that certainly doesn't mean that it's out there. I just haven't dug into it.
Thanks.
Okay. Any other questions on the thick ascending limbs potassium activity? All right. So then we can go a little bit further now. Now we're talking about our distal tubule and then our collecting duct. So here is where we have some things that are going to get things are going to start to get a bit more uh, interesting, and I'll say a little bit more confusing as well too. So now when we're starting to get distally, we have several transporters that are here. So we have our sodium chloride cotransporter. This is our thiazide sensitive transporter. So when we give hydrochlorothiazide as a diuretic, this is where it is working. Not a very potent diuretic because this is after we have the loop of Henle that's maintained our concentration gradient. This is where we are probably at only about maybe 10% of our sodium that's going to get reabsorbed in this site. So that's why it's not a very, very potent diuretic. But we have our thiazide transporter that is here. Again, everything is being driven by that sodium gradient that's being established by that basolateral sodium potassium ATPase. So here we still have our region of the outer medulla potassium channels, our ROMKs that are present. And as the proximal, well, let me say that differently, as our early distal tubule becomes our late distal tubule, it starts to develop different transporters that are present. And so there's different activities early versus late. Remember, when we're talking about the early distal tubule, so that is filtrate coming from the loop of Henle, the sodium potassium 2-chloride cotransporter is the main method, but there are other transporters that we haven't listed. But the filtrate at this level is quite dilute. So you have a hypotonic filtrate in your distal tubule because you have actively reabsorbed those molecules, and the thick ascending limb is impermeable to water. So you're just actively reabsorbing solutes rather than water. So things are actually quite dilute here. There's less solutes that are present. So we have some reabsorption of your mostly sodium here. Now potassium, this is where we start to get more potassium excretion rather than reabsorption. There's really not a luminal transporter for potassium reabsorption here. So here's where we're going to secrete potassium, and that's going to happen under normal circumstances. It's going to be as a result of our sodium and chloride that we're reabsorbing. We have in the distal, sorry, let me say that again, the late portion of the distal convoluted tubule, we have our epithelial sodium channels, our ENaCs. Remember, amiloride is the inhibitor of that. The ENaCs, we've discussed this previously, those are what become activated when we have various serine proteases that enter the tubular filtrate with glomerular disease. So things like plasminogen that gets cleaved by urokinase into plasmin, and plasmin activates this ENaC that will cause avid sodium reabsorption, and that causes volume expansion. That's the overfill mechanism for edema formation in nephrotic syndrome. So we've already gone over that, but just as a reminder and bringing it all together again. And because of the accumulation of sodium inside the cells, we're going to start to have some potassium that's going to get exchanged for it. And again, as the distal tubule becomes later, early distal tubule has some features suggestive of the loop of Henle's activity, and the late DCT is starting to act more like the collecting duct as well too. Now, what is really interesting here is that in the distal convoluted tubule, this is where we start to have the effects of aldo. Now, the aldo receptors are mostly in the collecting duct and the later portion of the DCT, but collectively that is referred to as the aldosterone sensitive distal nephron. So, that abbreviation is throughout the text a little bit, is basically talking about late DCT and collecting duct where there are aldo receptors that are present. So we're going to start to talk about aldo here. Now, what is really interesting is that in these late distal convoluted tubular cells, you start to have the presence of 11-beta hydroxysteroid dehydrogenase within the internal, within the intracellular environment. So that enzyme is present. And that enzyme is what metabolizes cortisol to cortisone. Now, cortisone doesn't have any efficacy, doesn't have any effect on the aldo receptor. Cortisol does, obviously not as much as aldo, but cortisol does have some cross reactivity, and it will stimulate the aldo receptor to some degree. But we have this 11-beta hydroxysteroid dehydrogenase that's going to turn off the activity of cortisol stimulating the aldo receptor by metabolizing it into cortisone, which is inactive. Now, this enzyme, 11-beta hydroxysteroid dehydrogenase, this enzyme is inactivated by, I think it's glyceritic acid, which is in licorice. So actual licorice, not just the black Twizzlers, but actual licorice has this glyceritic acid. I'm probably pronouncing that wrong, But this is how licorice can cause hypokalemia. So licorice, if you eat enough of it, it will inhibit this 11 beta hydroxysteroid dehydrogenase, meaning your normal circulating cortisol maintains its active binding to the aldo receptor, doesn't get converted to cortisone, so it's not inactivated, and it will give you what's called a syndrome of apparent mineralocorticoid excess, which just sounds awesome, right? So it's essentially like you have hyperaldosteronism, that you're going to have the aldo receptor that is stimulated, and then secondary to stimulation of aldosterone, we're going to get into this more in just another minute or two, it increases the activity of both the basolateral sodium potassium ATPase, it adds more ROMK channels, and you have greater potassium excretion into the lumen, and you'll get hypokalemia. You also, because you're reabsorbing more salt in the basolateral side, you're going to pull in more water and you get volume expansion. And so licorice can cause your patients to become hypertensive and hypokalemic, just like hyperaldosteronism could. And if you were to measure an aldo concentration on this human, because I'm going to imagine it's probably more of a human problem rather than a dog or a cat problem, you would see that this patient looks just like a hyperaldo patient, but their aldo concentrations are super low. And it's not driven by aldo. And that's why we call it this syndrome of apparent mineralocorticoid excess, which is cool. And unfortunately, people have actually died. There have been reports of people having cardiac arrest because they eat so much licorice that they develop severe hypokalemia associated with it. Just trying to, yep, those are my notes, but I didn't write the name of the acid. I think it's glyceric acid. Again, I'm probably pronouncing that incorrectly, but that is how we can see hypokalemia associated with licorice. Interestingly, there have been some studies in people with CKD, with end-stage kidney disease, where they're anuric and they're on dialysis, that are hyperkalemic, that have used licorice as a way to try to control their potassium. And they actually did so with modest success. And this enzyme is also present in the colon. You can really think of the colon as just kind of a big dumb nephron, and has many of the same receptors, and aldo can cause an increase in potassium elimination through the gut. So they've actually tried using licorice to control some hyperkalemia in people. I've never tried it in a dog or a cat, but I guess theoretically it remains a therapeutic option for us. Okay, so that's the distal tubule, and then we certainly get in down here into the collecting duct, and this is where we're going to have our intercalated cells and our principal cells that are involved here. Now, the intercalated cells, I always remember that by HI. So the intercalated cells are what's going to secrete hydrogen ions into the tubular lumen. So the intercalated cells, what's responsible for managing the acid-base status, secondary to being stimulated by aldosterone. And the principal cells are really more so where we have the above. This is where we're going to have our potassium excretion. So this is one of the really the main zone of what we talk about the effects of aldo. It's going to have more effects here on the principal and the intercalated cell than the late distal tubule, but remember, it still does in the late distal tubule too. But aldo is going to stimulate that basolateral ATPase. So we have more potassium that is accumulating. It's going to add those ROMKs. It adds this second transporter called a maxi-K, which has another name for it. I'm trying to see where I highlighted that. Also known as the large conductance channel. So you might see some papers call it the BK channel, but it's now mostly referred to as the maxi-K channel. So aldo will also stimulate that to be there. So aldo is going to put all these potassium efflux pumps on your luminal side, increase the basolateral ATPase, so we have a higher intracellular concentration of potassium, and we're going to get potassium to be excreted. Because that sodium potassium ATPase is more active, we are shuttling more sodium out of the intracellular space into the basolateral side, which is why we have sodium coming in through the ENaC. So that's how we're going to get sodium reabsorption. Water will follow that. So that's how we have volume expansion under the influences of aldo and potassium elimination. So that's going to be your potassium and sodium handling in the principal cell, and the intercalated cell, as I said, that's really just the dumping of hydrogen. So if you have a type 4 renal tubular acidosis, that's where you have either an absence or you have antagonization of the aldo receptor. So either no aldo, so this is the RTA we see in our patients who have Addison's, or you have resistance and you have an inability to excrete these hydrogen ions that will cause a normal anion gap metabolic acidosis. That's your type 4 RTA, which is the only RTA that is hyperkalemic. All the other type 1 and type 2 RTAs are hypokalemic. And if we go back up, why is a type 4 RTA hyperkalemic? Because we're also inhibiting the ability of aldo to stimulate that basolateral ATPase to put all these potassium efflux, the BK and the ROMK channels on the luminal side, so we can't get rid of the potassium. So I can't say I've ever diagnosed a type 4 RTA other than saying, oh, look at this Addisonian patient, and they have findings consistent with the type 4 RTA, but I still just call them Addisons. I can't say I've diagnosed that outside of a patient having Addisons. However, it is possible that you can see type 4 RTAs in patients with CKD that have maybe diminished tubular cell functions. They might have some degree of aldo resistance. So to some degree, the hyperkalemia that can be seen with later stage CKD could in part be due to a type 4 RTA. I've seen that in one patient actually that had it was a healthy cat that had a dental and developed an AKI secondary to meloxicam, and then developed a significant hyperkalemia, and it was a non-anion gap metabolic acidosis that was pretty persistent. And that cat actually did really well. We could just give it a mg per kg of Lasix, and that was enough to cause enough potassium to be excreted upstream that it didn't matter what was happening down here. But I think that's probably one of the only cases that I really worked up to show that they did have a type 4 aldo independent RTA. We measured that cat's aldo concentrations and they were through the roof. So it wasn't an aldo deficiency, it was aldo resistance. And, it's on my pile of case reports to eventually write up. But yeah, that's the only one that I've seen. I don't know if other people have seen and really diagnosed a type 4 RTA, but that's why we're going to get it from what's happening here in the intercalated cell. Questions or comments for where we're at here?
JD,
Okay,
the only
yeah, go
comment I
ahead.
wanted to make was the potassium chloride um, illustrated in kind of that orangish reddish color there.
Mhm.
Uh, that doesn't show up in many textbooks, and I don't have any idea of the magnitude of the amount of potassium excretion by that method in the
Yeah,
late distal convoluted tubule and the principal cell. But I teach physiology. I don't include that at all. Um,
yeah. I, I and it's interesting because when you look here, unlike in the early DCT where you have this thiazide sensitive sodium chloride co-transporter, we don't really have an easy way for sodium to enter this, chloride to enter the cell. So we have this same efflux pump like we do in the DCT, but I don't know the significance of it. I agree with you, Larry, because we don't have this chloride entry pathway that we did in the DCT. So I agree. I'm not 100% sure of that mechanism, other than it just being another way for potassium to be eliminated. But it seems like even if you're getting rid of that intracellular chloride, it just gets reabsorbed paracellularly. So it might not have a major impact overall.
Yeah, thanks.
Yeah. Uh, so then one of the other things that I think is really interesting is they talk about this tubular flow rate, and that under states of high or low tubular flow, it might actually flip how your patient, how the distal convolute tubule and the collecting duct are active. And they say that it's actually due to intracellular calcium concentrations that are modified because of the flow rate, and it has to do with this central cilium. And I wanted to bring up, when I read this part in the paper, it brought me back to this image that I love, and this I think is has been put in Brenner and Rector. But this is the collecting duct. This is the rabbit collecting duct, and we can see every one of these cells that has this large central cilia, this big hair that's sticking off in the center of it. That is a principal cell, and the cells that are next to it that lack it, that's an intercalated cell. So I just thought it was cool to actually, I've seen this image before, and when I read about that central cilia, I was like, oh my gosh, awesome. That might be what, I'm sure it might do other things, but it's actually pretty cool. It's this flow sensor. So as you have tons of flow going down the nephron, and that cilia is getting pushed and it's getting pulled down because of that flow, that is going to activate these calcium channels, and you'll have intracellular calcium that's going to accumulate, and that's going to affect the number of BK channels and ROMK channels that might be there. And I thought that that was just a pretty cool mechanism of sensing how much flow um, that might be there. So I wanted to share that image with everybody. They go in a little bit deeper about the role of calcium and its transporters, and I think it's interesting, but it also gets pretty heavy, and I think we have about 8 minutes left. Um, the other things I wanted to bring up is I think most veterinarians have not heard of Bartter syndrome, and they bring it up here. So Bartter syndrome is a mutation in the ROMK, and it's loss of function. So that means it's anytime you have a loss of function mutation, it almost always is going to be a recessive inherited trait, because you're going to need to have a double knockout to see it. But it's going to prevent potassium leaving the cell through that ROMK, and it's going to prevent that recycling. So close your eyes, don't puke as I run up here again. So we're back into thick ascending limb. So people with Barter's have a mutation, so they don't have a functional ROMK. So you can't have the potassium that's going to leave the cell, meaning that you're not going to have potassium available for the sodium potassium 2 chloride, and it essentially just shuts down this co-transporter. So people with Bartter syndrome, they are living life as if they are chronically taking Lasix. It is essentially a syndrome that's going to mimic a diuretic administration, a loop diuretic. So I I absolutely think that we have patients that likely have these genetic causes of hyperkalemia, of hypertension, and I just don't think we are sufficient enough, sophisticated enough to know where those mutations might be in dogs and cats. But unless we have things like Bartter syndrome and Little syndrome and Gordon syndrome all on our radar, and we've heard about it, we're never going to actually diagnose it if it's just not something that we've ever heard of. So that's disease is something that's recognized very young in the age of people, and there's a strong family history of the hypokalemia going along with it. But just wanted to point that out, that I suspect that that is a disease that we do see, but is not yet been reported in dogs or cats, to the best of my knowledge.
Do you
Uh,
think with, sorry,
Go
do you
ahead.
think with the, those dogs that are chronically PUPD and no cause can be found, maybe some of those should be considered as that?
I think I do think that there's probably a good a good chance that some of those patients, one of those patients somewhere, is going to have something. These diseases are not common, even amongst nephrologists. They might manage, one or two actively throughout their clinic. So it's not as if these are mutations that are running around with high affinity, but it's just, yeah, we're never going to diagnose it if it's not on our radar. And if it's not on our radar, then maybe we haven't even looked to see if dogs could potentially have those same mutations, and it's just an unevaluated differential. So I think it's possible. So maybe in those dogs with significant PPD that are failing to have an easy diagnostic answer, maybe we need to do some genome screening on them to see how that might compare to a non-PUPD dog. But I am way out of my element when it comes to thinking of any type of genome screening or testing for any of these mutations. That's way too smart for what my mediocre brain can handle. But sure, I think that some of those patients, they could have it, and I don't think we're going to diagnose it unless we start looking for
Yeah, cool.
it. Okay, and then the last thing that I wanted to bring up, and you can see I almost highlighted this entire column, but I think it's a very cool concept of how aldo can regulate potassium and sodium differently. And that's what's called the aldosterone paradox. That even though aldo is going to be acting on its same receptor, how do we have, in situations where we have volume depletion and activation of the RAS and the aldo activity that's increased, and sodium and water that's being reabsorbed, why aren't we seeing those patients who develop significant hypokalemia associated with it? And how are we able to excrete the potassium after we eat a meal? Because recognize, with every meal that you eat, it contains a lethal quantity of potassium. If that potassium would not get shuttled inside the cells immediately, you would die. That's how tightly regulated our potassium concentration is. So it's this aldo paradox that allows us to think of it being independent control of sodium versus potassium. It has a lot to do with that tubular flow rate. There is now a lot of emphasis placed on this wink uh, channels, and I love how they came up with the name. So that stands for with no lysine. K is the abbreviation for lysine for the amino acid code. So with no lysine, WNK. So the Wink kinases are heavily involved in this in terms of just area that's getting researched. And essentially, we have these two different scenarios. So if we start with the first one, this is our one that we're probably more familiar with. We have decreased effective arterial blood volume, and you can call that decreased effective circulating volume, renal hypoperfusion. You're going to have increased proximal sodium reabsorption, and that's going to be somewhat through catecholamines. That's also going to be through the effective Ang2 that's going to do that. And that's going to lead to less sodium being delivered into the distal nephron. There's going to be less water in the distal nephron because you're trying to reabsorb everything. It also will lead to increased renin. So remember that decrease in your distal sodium delivery is going to be accompanied by a decrease in the distal chloride delivery. And that the macula densa will see that low intraluminal chloride, and that's what causes it to release renin. And so we have renin that will cleave angiotensinogen into Ang1, Ang1 becomes Ang2, and leads to increased aldo. And that should cause potassium wasting. But because we have very low flow, because we have little sodium and therefore little water getting to the distal tubule, we're going to have um, differences in the expression. And some of that's through the Wnt kinase, some of it's through the action of that cilia sensing the flow of the tubular filtrate across, and that's going to change the location of your ROMK and your BK channels that are there. So this is how we can have all of this aldo that's going to be there and effectively cause more sodium reabsorption, but it will minimize potassium wasting. We don't become hypokalemic in that scenario. When we have increased effective circulating volume, then we have less salt that's going to be reabsorbed proximally. You're going to have increased distal delivery of that sodium, and that will cause increased tubular flow. And that should cause more potassium wasting because now that cilia is activated, we're gonna have more ROMK, we're gonna have more BK channels that are put onto the lumen, and that should cause renal potassium wasting. But we also have less renin because we have more sodium and chloride in the distal tubule, therefore less aldo. And so aldo is actually going to stimulate that uh, aldo is not going to stimulate that basolateral sodium potassium ATPase. So in both of those scenarios, because mostly due to the tubular flow rate, even though we have aldo that's going to be affecting things, the effect of your renal potassium wasting in states of increased and decreased effective circulating volume doesn't typically result in potassium abnormalities. So it's a pretty interesting way that the body is able to maintain volume without causing significant derangements in potassium. And this is this is obviously an abbreviation. You folks have read it, but there's so many details that are there. But when you read one step at a time, it makes a really nice story. And it surprisingly has to do a lot with that flow rate, which I don't think that I certainly didn't put a lot of emphasis on in my training. And here is some information on that here. So here's where we're having hypovolemia causing Ang2 and aldo, and aldo is going to cause more of these sodium channels that are going to be there. You're going to have potassium elimination, but you're having low tubular flow rate. Difference than when you have high tubular flow rates, which could cause the or maybe this is actually just, and yeah, I think this is actually just under states of hypertension as well. Sorry, hyperkalemia. That's going to cause aldo to become stimulated. So aldo is a really fascinating hormone because it independently has the ability, even though it's the same intracellular mechanisms, to regulate sodium and potassium. And it has to do with the movement of water past the luminal surface of those cells, which is really, in most patients, driven by the amount of sodium that's there. So the more sodium you deliver to the distal nephron, the higher the flow rate you're going to have, with the exception of things like diabetes mellitus or a patient just got a big dose of mannitol or something like that. So I thought overall the article did a good job of trying to explain and highlight how those two things are independent. I probably have done a very poor job of doing it, but I thought that was just a really nice review of the physiology of volume control versus potassium control that's there. Okay, let me pause there and see if there are any questions because we're kind of at the end of the hour. I think we've covered most of what I thought were the big things, but let me see if there are any questions that we can try to get through before we bring this session to a close.
I thought you did a great job clarifying things. I just want to make a quick comment about flow rate. We think about that with urea absorption and how important it is with that passive reabsorption. And for everything else, I've always kind of assumed it's the same thing. It's just not getting reabsorbed because of the faster flow. And of course, it's more complicated. So that was
Yeah, I totally
interesting.
agree. Right, my brain wants it to be simple. More H plus going in, K plus goes out. Fast flow rate, not enough time for reabsorption. And no, it comes down to these cilia flapping, not in the wind, but flapping in the water and causing intracellular calcium to accumulate, these transporters to go to the luminal surface. And even though that is so much harder for my brain to understand, it is so awesome. I love that physiology. It is so cool, but I will remember 4% of this tomorrow after I finish,
Yeah.
kind of going over all this. So I'll have to keep going over it again and again because I think it's a lot easier to stick with those more simplistic mechanisms, which are not totally wrong, but also I think don't give the complexity of renal physiology quite the justice that it deserves. But man, it's brain work to understand everything and to get through it. But I thought this article was actually really nice, and I love the way that it was written. It just didn't take anything for granted that the readers were knew everything that was already there. It really laid it out in a simple way. So I thought it was a really nice review of potassium physiology, and hopefully people didn't find it to be too much of a tortuous read.
It is cool. It's also sad that we can't make it more simplistic. Although sometimes, even with a complicated mechanism behind it, I'll still tell students, think of it this way so they'll remember it,
Oh, for
at least
sure.
the
Yeah,
basics.
absolutely. And I think this is probably the difference of where we really would want our nephrology fellows to, be, they know what an ENaC is, and they know what a ROMK is, and they know that there's a lot more detailed mechanisms, even if they can't remember everything. And yeah, and I think those are going to be the people who might diagnose the first dog with Bartter syndrome or the first cat with Gordon syndrome or something like that because it's on their radar. Where I don't think I did not learn any of those diseases during my medicine residency for sure. I didn't teach them to my residents when I was faculty. It was just not on my radar until I just continued kind of these deep dives into human nephrology. So I hoped that we're going to pull some of those diseases out and we'll probably get more sophisticated in our diagnoses and maybe help some patients that we otherwise wouldn't have had a good understanding for what was causing their disease.
Yeah. Thank you.
Awesome. All right. Any other questions, everybody, before we bring this session to a close? All right. Well, this is our last session of the year. I will send out another registration just because I haven't scheduled any of our meetings for 2023. So I will send everybody an email with a registration link so that you can register for the 2023 sessions. And then there will be a new link to actually join them. So yeah, I hope everyone has a wonderful holiday. I will come up with some articles for our next topic and I will send them out. I will get this video uploaded onto our YouTube page probably in the next day or two, and you can find it linked on the Renal Fellows page on the ASPNU website. So always great seeing everybody. Hope everyone is well, and I will talk to you soon.
Happy holidays.